other people who have come to attend this conference. Now, today, what I'm going to talk about is the application of the enzymes and the whole cells for the production of the drugs and the drug intermediates. Now, this, we will not talk something about the plant or something about some amount of the plant things, not animal cells, but on the microbes only, and how we have used the microbes for the production of different useful technologies through research that I can show it to you. Now, you can see that our areas of the focus of the following: the production of the various secondary metabolites. And these secondary metabolites, starting from the rifampicin or rifamide antibiotic, which you know very well, which comes directly from the fermentation, and then rifamycin oxidase, which converts the rifamycin B to rifamycin S. And the rifamycin S is the starting compound for the production of the rifamide. That technology has been given to the industry. And then tacrolimus, mycophenolic acid, citric acid, all the different types of the drugs, you can say. And then something about the production of the different types of the interferon beta and interferon alpha by recombinant fermentation technology, where you can produce it in 7-liter reactor and then purify it, and you can check it through the Western blot and some other things. We also work a lot of the nanoparticle synthesis by the microbes or by the plant materials. And we use that nanobiocatalyst, we use those nanoparticles for the production of the nanobiocatalyst, and those nanobiocatalysts can convert very easily the different types of the racemic drugs. All you know very well, the drug contains the two components. One is the E tumor and the dis tumor, and which you want to separate it out by the chemical methods, which are very chemical technology, which is very well known. But now, because of the green chemistry, you have to do this biochemically. So we have tried through the free cells, we have tried with the different types of your immobilized, conventional immobilized methods, but then we have shifted to the nanobiocatalyst, which is a huge, tremendous potential for the production of the different types of materials. And then those nanomaterials were taken for the production of the PDT, photodynamic therapy. And then PTN, lots of PTNs have been produced, and the PTNs were used for the cell killing, Pseudomonas or E. coli or your Carbuliria albuminata, different types of the materials. And then we did lots of works on the in vitro assays for the cancer. Actually, what we do, because the biology group, there are chemistry group, they can synthesize the chemistry, all the substrate analogs and all these things they can synthesize, but the assay is very difficult, biological assays. So those assay parts have been done, and some of the your NCEs, new chemical entities, have been patented, and also we have published a lot. So if you can see that we do generally practice, it is a wonderful ways to do it, that we do culture the various biocatalysts from the nature, and the culture collection laboratories like microbial type of culture collection laboratories, and so many others, ATCC or maybe in NCCS Pune. And then we optimize the growth and the production of the microorganisms in the sec flask. Then we do it in a little bit bigger one, that is in the reactor. There is a 5-liter reactor, 2-liter reactors. We purify the enzyme, we characterize the enzyme, and then we do the biocatalysis. And then we synthesize the nanoparticles with the biological materials, and then we do the different types of kinetic analysis. Now, this kinetics completely changed. We do the thermodynamics and the kinetics analysis of this biocatalyst, and we find out different types of kinetic parameters. You see, this is wonderful, although it is a very old work, but its significance is very good, because you can see how you are going to produce the rifamycin B. Rifamycin, all we know very well, rifamide or rifampicin is a very broad spectrum antibiotic, and that is produced by Nocardia mediterranea. This Nocardia mediterranea, when it produces, although fermentation is very difficult, but you can biologically convert it by rifamycin oxidase. So whatever the rifamycin B is produced, that you can convert it to rifamycin O and then to hydrolysis rifamycin S. And in order to do so by Carbuliria albuminata, you do different types of this effect of elation, effect of agitation, effect of all these things. Why? Now we are doing it for increasing the productivity of the microorganisms, productivity of the enzymes, titer of the enzymes. So when you get the more the enzymes, rate of reaction is more. Suppose the rate of reaction is more, then you can get the total productivity of the whole substrate converted to the product is much better. And in that process, what we did the biotransformations. I know with a little bit of biotechnology background, this will be much better to realize, much better to understand. But you can see, I'll show you after 2-3 slides that how we have transferred this technology to the loop-in chemicals that time. We have IDPL, Rishikesh, that time also we have given this technology. Both the technology were given and the institute earned a lot of money on this one. But the basic works we did it, we published the things, we have patented the things, and also we have given the technology. Now if you can see some of the chemical engineering aspects also you have to do it, that is biotransformations with the growing cells, biotransformations with the resting cells, biotransformations with the resting cells, feeding with the glucose, and all these experiments you can see how we have done it, and then we have published simultaneously also. And if you see this graph, this table says that how the agitation and aeration helps for the biotransformations. But anyway, I will not explain much on this one. These are the things where we can see that a particular cell mass, Nocardia mediterraneae or maybe E. coli, which is not producing the rifamycin, how much amount of the rifamycins are being absorbed on the cell surface, that we have found out very nicely through this experiment. Then you can see, after all, when the whole your scientific things have been done, then the institute, it was the institute order or the director's order that you have to produce the technology and we will have to give to you can see the technology was produced in LKB fermenter, 6.2 liter and 17.2 liter. Although these are the fermenters which are available from the UK. And then we did it, and ultimately you can see that technology was transferred to the IDPL Rishikesh and the loop-in chemicals laboratory simultaneously. So this was a complete work, like this alcohol production also, although alcohol does not come under this purview, but alcohol production from the molasses, if you see, the technology was developed from the batch culture to the continuous culture. Anyway, now if you see another technology which you did a lot of work, and you know and how the streptokinase works. If you use streptokinase, actually streptokinase is an inactive molecule. But when the streptokinase it reacts with the plasminogen, then the complex which forms, that is the activated streptokinase. And this activated streptokinase, it converts the plasminogen to plasmin. And plasmin is responsible for making the fibrin insoluble to fibrin soluble. And that is the way the heart attack is. It works like this. And when this organism was grown, Streptomyces equisimilis, it was grown in the sec flask, it was grown in the 7-liter fermenter, and the whole technology was transferred to the 1,500-liter reactor. Initially 150-liter reactor and then 1,500 reactor. And the technology was given to, you can see the Messrs. Cadila Healthcare, Ahmedabad. So the whole technology was developed within 1.5 years. 2 years it took it, and we published the papers and all these things. But this is the things from the it is not genetic engineering. It is a wild type. But after that, all types of the genetic engineering, this is site directed and all these things came into market. This technology was given to 2 to 3 companies. And then another one, it is a phytase. You can say phytase, all you know very well, that any feed, if you want to make it, there are 700 feed makers in our country, making tons and tons of the feeds. And these feeds contains the phytic acid. And this phytic acid, as you know the phytic acid, you can see all their very charged groups. So all their phosphate groups. So when it goes to the stomach, then it can chelate the metal ions. So that is a very bad thing. So what will happen, per ton of your feed, if you add 200 gram of phytase, so that whole phytase can act on and with the agitation and aeration a little bit, then they can take out the phosphorus groups from this phytic acid. So inositol, that is the inositol monophosphate, diphosphate, triphosphate, pentaphosphate, hexaphosphate. Hexaphosphate is phytic acid. So what you can do, you can get the different types of your mono, di, tri, tetra, you can take it out through the different types of the columns. And we did it in a very nice way, patented it and given this technology to a South-based company. And you can see these are all the reactor studies. Here you can see how the phytic acid is hydrolyzed in presence of the feed. So this is a real experiment with the feed. If the feed is taken from the Central Poultry Control Board, Central Poultry Board from Chandigarh. And you can see these are all the activities which generally a mass transfer coefficient, heat transfer coefficient in any, when you are going to develop any technology, you have to do this. Then you can see these are all the enzymes are purified and all these things. Ultimately these are all your LCMS, and you can see how the fragments were all differentiated out. And we did this myoinositol hexaphosphate. There is a product inhibition, there is a substrate inhibition, And substrate is a competitive substrate inhibition. A substrate analog was taken that is called MIHS. MIHS is the bioinositol hexasulfate. So when we have added the bioinositol hexasulfate here, we can see a competitive inhibition. And if you extend the graph, you can see the Vmax remains the same, but the Km increases. So when the Km increases, Vmax remains the same. So we modeled it and we have found out that yes, these are the enzymes which can be inhibited, and you can find out the inhibition kinetics very well. And this enzyme is still, there are 5 or 6 companies in India which are making this phytase. They make the cocktail of enzymes and give it to different types of your feed makers. Next, we have we did a lot of work on the arginine deaminase. All you know very well that these cancerous cells, they do not have this arginine, they cannot get the arginine, leucine, lysine, and then asparagine, all these things. But through the blood circulation as they take it, and that is the normal mechanisms by which the cancer cells, they are, if you put the asparaginase, if you put the methionase, if you put the arginine deaminase, they can remove all their precursors, arginine and all these things, and then the cells die. So one of the student, he worked a lot on this one, arginine deaminase from the Pseudomonas putida, and we scaled it up to 10 liter reactor. And although the technology has not been given to, so far it was not given to anybody, but we have to do a lot of work on this. And methionase and the arginine deaminase, these two enzymes are, asparaginase of course it is there, the technology is already there. So asparaginase, you don't have to do anything. And then we have different types of the tacrolimus, and then tacrolimus has been produced by this one, and another is your production of the mycophenolic acids. The mycophenolic acids and the tacrolimus, these two drugs, although they are very known drugs, but you can see that there is no technology in our country. All we get it from your China and all this. There are few companies which are making it, but they generally do it formulations. So we had an organism which produces around 8 grams per liter of, 4 grams per liter of the mycophenolic acid by these organisms. Then we have this interferon beta or interferon alpha. These interferon beta and interferon alpha, with one of my colleagues, Abhay Pandey, we have cloned the we have cloned the gene, interferon beta alpha and interferon gamma, and then we have characterized it through the Western blot, and we have seen, but production is very less. We are also still working on this one to make a very good technology. This, again you can see the production of the succinic acid. All you know why the succinic acid is very much required, because succinic acid is the Tamiflu. Tamiflu ka succinic acid is the precursor material. We have the AROK knockout. If you see the biosynthetic pathways, AROK knockout means the synthesis of the succinic acid will be there, and then succinic acid will not be degraded from that. So AROK knockout mutant we created through Bacillus megaterium, and that Bacillus megaterium was grown, and then lots of your kinetics we did it, and we published several papers on this one. Then, what happened? If you see, from the 1962 onwards, there was a enantiosuitive synthesis of the drugs and the drug intermediates using the different types of the enzymes that is going on, because there is a very big tragedy, and that due to that tragedy, what is happening, you can see this one. So what is happening, your thalidomide reaction. The thalidomide R and S, when you give the mixture, one is your teratogenic cancer causing material, and another is it reduces the pain of the pregnant woman. So what is happening, these are the results of this one. So from 1992, USA FDA has promulgated a rule that whenever the drug will come to the market, okay, so that will have to be your enantiomeric. So what we did, we took the nitrilase, amidase, penicillin acylase, hydantoinase, and some of the enzymes. We have taken it, immobilized it, and again we have put it into this one, and we can see that how we have made it, separated all the things. We have cloned it out, and we have seen the CLIA, the cross-linked enzyme aggregates, we have made it, and these cross-linked enzyme aggregates have separated out many other things. Anyway, so doluxetine is also another drug which is a cardiovascular drug. This doluxetine we have enzymatically synthesized from the raw materials. You can see the different reactions and other things. This is also another Geotricum candidum. This can do a lots of your oxidoreductase reactions and all these things. And then lipase-mediated reactions, you can see this is actually different. CDPP. CDPP is the drug which is that propanol. Yeah. So this is the MPGM. MPGM is a diltiazem. Diltiazem is so far is not produced in our country. Diltiazem, the precursors MPGM, is taken from the China, and we formulate it. So diltiazem, what is happening? Diltiazem, the MPGM we have isolated and we have separated out by the enzymatic reactions. So like this, we have done there are lots of adrenergic blocking agents like your chemoenzymatic synthesis, encyprazine, bunitrolol, esmolol, acetyl-butanol, all different types of beta blockers. We worked a lot and enzymatic separations. I will make it a very because the time is not much. Then you can see nanomaterials we have made it. You can see this is that your silver nitrate solution. This is an enzymatic solution. Both you mixed and you can get the nanomaterials. These nanomaterials we have used it, and all the nanomaterials we have made it in a little bit further quantity, and we have used it for the antioxidant assay. And you can see the DPPH, ABTS and MTT assay, all the assays were done. The take-home message is that when that nanomaterial is impregnated with different types of the plant material, they are much better antioxidant. If you take the sigma, you purchase from the sigma, these nanometals and all these things, they are not very good antioxidant. They are not antioxidant at all. So this is like the nitrilase. Here also did the modeling of the lipase reactions. Here you can see, and different types of here we have seen that how the secondary structures of the molecular secondary structure changes and all these things. Here also it is the nanobiocatalytic reactions, and these nanobiocatalytic reactions are done by the lipase reactions. Here also actually different types of your chemicals we have taken, and thermostability has increased. You can see this is the graph where the thermostability and different types of your thermodynamic parameters have been cleared out. Here also we have done the papers on the organic, inorganic, hybrid nanoflavor. This has come to the market nowadays. It will coming very soon. That is the hybrid molecule. So hybrid molecule, we can do a lots of your biotransformations to the hybrid molecules. And this is also another nanopolyethylene nanofibers. This is, now the photo, I will just 1 minute. This is phototheranostics agents. You can see due to the EPR, that is that enhanced permeability retention factor, all these advantage we get in for making any types of your PDTA. And we have taken the rose bengal, rhodamine B and porphyrin. Rose bengal, rhodamine B you can buy it from the market, but porphyrins were synthesized. Different types of the body B and porphyrin were synthesized. We have put it into the nanoparticles and we have used it. You can see the bio-inspired theranostic agents. These bio-inspired theranostic agents have been put it, and the different reactions we have done it. And ultimately, we have taken the same term and we have seen it through the CLS, confocal your microscope. Now this confocal laser microscope, we have seen that you can see the cell killing is high, very high cell killing. Propedium iodide, you can see the cell killing. And this cell killing also we have done in this case. In this case also we have done, we have seen it through the TEM and SEM that how the cells are killed and how the cells are, all the brittle cells are coming out. So that means our systems is working with the 80 to 90% loading of rhodamine B and your porphyrins. So this is another work, but this is the DNA. We did a lot of basic work on the kinetics of the topoisomerase 1 and topoisomerase 2. And we have published a half dozen of paper, and also we patented it for the new NCs. Anyway, so this is the conclusions which I can draw from this one, whatever I have told. That with the quick identification or active and unselective nitrilase we have. So basically what we have done, we have lots of enzymes from the microbes. We have isolated the enzymes, we have isolated the enzymes, purified it, did the kinetics, thermodynamic studies, and we have used that enzymes for the immobilization. And of those catalysts, as a nanobiocatalyst, we have used it for the separation of the different types of your enantiomers. And then we have, although our PTNs have solved the good antimicrobial activity, but they also have some of the problems. Problems are that loading is still less. So we are trying to make it maximum loading into the, so we have come to the gelatin nanoparticle. So gelatin nanoparticles will be, and we are working on this one, it is given. So these are all my actually students. The students are the, they have done, you can see, the even BANERJEE, the Sara Singh, these all, and the Satyendra knows all the people, and this is the group where this is NIPER, and this is some part of it where all my students are working, and these are the products of those students. Actually, I have not done much, but my students have done all the things. Thank you very much.